Hi everyone, I'm Brittany, and this summer I've been working with Travis and Joe looking at the bacterium Brevibacillus laterosporus. So Brevibacillus laterosporus is a spore-forming bacteria which is toxic to a range of insects including the diamondback moth, the larvae of which feed on brassica crops causing extensive damage. The mechanism for its toxicity is not yet well understood, but a biopesticide is being developed using the bacterium. We've explored the effect of culturing two strains 1821 and 1951 together, as well as evaluating the effect of culturing both strains in media with different sugars and nitrogen sources. So the aim of this project was to explore how to optimize toxin expression under different conditions using bioassays with diamondback moth larvae. So we applied treatments to the larvae using a drenching method, and as you can see up here, this is a healthy larva compared to one that has been killed by the toxin. So previous research has indicated that growing the two strains together can result in a greater number of cells than culturing the strains alone. However, in our first experiment where we evaluated culturing them in a 1-to-1 ratio, we found that low inoculum 1951A cultured alone resulted in the highest CFU counts, the highest mortality. And also looking at the SDS page protein profile, this treatment shows a greater amount of protein expression than the other treatments. Next, we looked at culturing the strains together in a 1-to-3 ratio. And we found that 1951 cultured alone at 1 mil resulted in a higher CFU count than the dual cultures. And in the bioassay, we found that the 1-to-3 dual culture of 250 microliters of 1821 and 750 microliters of 1951 had the highest mortality rate. Next, we used PCR to determine the strain ratio in the dual cultures. We couldn't just use looking at the colonies alone because they have identical colony morphology. So we selected 100 colonies off the dual culture plates and used two primers, the 16S universal primer and the Cry35 primer that is specific to 1951. So therefore, one band indicates that the colony is 1821 and two bands indicate that it is 1951. So analysing the 2 mil 1-to-1 dual culture, we found that 99% of the colonies were 1821. And looking at the 1-to-3 dual culture, we found that 97% of the colonies were 1821. So our results show that 1821 outcompetes 1951 in culture, even though the two strains are genetically very similar. And 1821 does not usually grow as well as 1951 due to colony collapse, so dual culturing may not be beneficial for maximising toxicity. We are currently preparing to do the same analysis for this treatment up here, which resulted in the highest mortality. Next, we looked at culturing both strains with the sugars glucose, fructose, lactose, maltose, raffinose, and sucrose. And we found that both strains grew the best in lactose, but they showed differing responses in toxicity, with 1951 having the highest toxicity in glucose and maltose, and 1821 being most toxic in lactose. So a higher level of growth does not necessarily translate to increased toxicity, and this illustrates how the strains can behave very differently. So these results suggest that culturing Brevibacillus with sugars other than glucose should be further explored, and the two strains may have different media requirements. And next, we cultured both strains in normal MLB media and nitrogen-rich MLB media, supplemented with ammonium nitrate and ammonium chloride. So again, the strains behave very differently under these conditions, but the differences in growth between the two media were very small. The SDS page protein profile shows little difference in the protein profiles between the media types, with original MLB being on the left and the nitrogen-enriched media being on the right. However, this clearly illustrates the difference in protein profiles between the two strains. with 1821 here and 1951 here. Thank you for listening, and thank you to Travis and Joe for all your support.
Questions for Brittney?
Yes.
Really interesting response to different sugars. I'm wondering if you had any sort of ideas about why that is, why the sugar toxicity.
We're not too sure because the toxin hasn't been identified yet, so we're not sure what the metabolic pathways are that result in the toxicity. So that's definitely something to be further explored.
Talk about the competition between the strains. What form does that competition take? Is it for resources? You find that you start them off on an equal 50-50 ratio, and then you've got a different ratio subsequently. So what's driving that?
We think it could be an antibacterial molecule, perhaps a protein, because there's been evidence in previous work that 1821 has the ability to produce something that excludes 1951.
Okay, that's fascinating. Thank you very much.
Thank you.